Hello, everyone. I'm Shravan Kannan, and today I'll be talking about a gene therapy for Lynch syndrome using CRISPR-Cas9 nickase. Lynch syndrome is a hereditary disorder associated with a cancer of the colon and other organs, as you can see here. However, the most prevalent and debilitating disorder within Lynch syndrome is hereditary non-polyposis colorectal cancer, also known as HNPCC. This is caused by mutations in genes involving in DNA mismatch repair. And as you can see in this diagram here, you can tell clearly with MLH1, for instance, which is involved with PMS2 to induce single-strand breaks, and MSH2, which forms complexes with MSH6 to provide stability for MSH6 to recognize mismatches. There are no cures present for HNPCC and Lynch syndrome. However, current management involves prophylactically surgical colectomy or the removal of the colon. This, however, is a very highly morbid and risky procedure, which leads to risk of bleeding, infection, and damage to surrounding structures. It can also lead to a lifelong dependence on parenteral or intravenous nutrition from a young age, and thus increased risk of infection as well. On a side note, Lynch syndrome is also considered to be more prevalent than the BRCA mutations that are responsible for breast cancer, as you can see in the statistics here. And every day, approximately 39 people are born in the US with Lynch syndrome. So this leads me to my new solution of a CRISPR-Cas9 nickase-directed genetic therapy to address the mutations that are responsible for Lynch syndrome. So first, I talk about a background on the CRISPR-Cas9 nickase system. The function of the system is that it's an RNA-guided molecular tool for gene editing, of which it has three components: the Cas9 nickase and the two guide RNAs, followed by a template. So the Cas9 nickase is a protein that initiates single-strand cuts in the DNA, as you can see in the diagram here. And the guide RNAs are 20 nucleotide sequences complementary to the desired genomic locus. And the PAM sequences are 3 nucleotide sequences that Cas9 recognizes, as you can see over here in the purple part of the sequence. And this is where what is also referred to as the Cas9 nickase binding domain. The template sequence contains the canonical sequence or the original sequence, and this can be used to replicate DNA to replace the mutated copies of DNA. So this leads me to my repair mechanisms. The repair mechanisms are non-homologous end joining and HDR. The difference between these two is that both of them cut out damaged DNA, but however, HDR replaces it. And they replace it with a precise edit by making reference of a template. On the other end, non-homologous end joining cuts out the damaged DNA and merges the ends together, which can lead to insertion, deletion, and frameshift mutations. Now to my experiment methods, followed by proof of concept experiments. So first, what I did was I mapped out mutation clusters. But to do this, I obtained the genomic sequence of MLH1 and MSH2 from the UCSC genome browser. Mutations that resulted in a loss of function or had a clinical association with HNPCC were queried from the UniProt-KB database and are mapped onto the structural domains of MLH1 and MSH2, as you'll see in the next slide. In this slide, you can see clearly how the mutations were mapped. All the red rectangles here are the mutation locations, and the other big rectangles at the bottom here are the deletion locations. All these triangles here represent, in this sense, the different types of mutations that are present and their increased prevalence. This was followed by guide RNA design and validation, of which 4 guide RNAs were designed, flanking the ATPase binding domain of MLH1 and the mut-L homolog interaction domain of MSH2. And the reason why these 2 domains were chosen was because here's where the mutations were the most concentrated for clinical association with HNPCC, as you'll see in the next slide. The genomic sequence was also searched for protospaced or adjacent motif sequences, and the guide RNA was assessed to ensure that it had an 80% GC content, and it would be verified by Cas and Offfinder, BLAST and Cas Offfinder for off-target binding. And as you can see in this diagram here, you can tell clearly where the guide RNAs are located for MLH1 as well as for MSH2. So now to the molecular cloning of IDLV packaging, plasmid containing guide RNA and Cas9N, which is also the beginning of my proof of concept experiments. So as you can see here, IDLVs first are lentiviruses with no replicative capability, but a high transduction efficacy and a broad tropism. What this means is these viruses are the types of viruses that can infect various types of cells, but are unable to replicate within cells. And this leads me to the components that I had within each IDLV. The first, the PLNT CRISPR V2 plasmid, the guide RNA sequence, and the Cas9N. First, we'll talk about the gRNA sequence, which is introduced as annealed oligos. And these are the different types of annealed oligos that I had to create, of where this is the gRNA sequence, followed by the annealed oligos, which were created using BBS1 overhangs at the end. Then in the pLNT CRISPR V2 plasmid, as you can see here, you can tell clearly the different types of elements that were needed for the IDLV itself, followed by the Cas9 nickase, and then the BBS1 overhangs and the guide RNA. Now to the creation of DNA template for homology-directed repair. As you can see in this diagram here, the original DNA template was around 400 base pairs, but using PCR, there was restriction enzyme ends were added, specifically the SAP1 overhangs. And these SAP1 overhangs were introduced via PCR. And using restriction enzymes to digest them, overhang sticky ends were created, as you see with these overlaps here. The same thing was done with the PSPAX2 plasmid backbone to create recombinant DNA. The reason why the total length is 816 base pairs for MLH1, 813 base pairs for MSH2 is because of the 200 base pair homology arms flanked to create HDR. Now, 2 template plasmids for MLH1 and MSH2 and the 2 Cas9N plasmids were injected into the media of human embryonic kidney 293 cells at 80% confluency. And after 24 hours, the supernatant would be collected, verified, and concentrated into pellets. Now to the theoretical proof of concept experiments. So first, I confirmed the cloning of the template plasmid, and for this, I've digested both the backbone and the one with the template sequence by making 2 restriction enzymes, and I ran them on, and I did them. They would run them theoretically on our agarose gel. Now you can see there's a 400 base pair difference on these bottom 2 bands. That is because of the presence of a template. In terms of HDR efficacy, the ddPCR technique will be used. And here you can see clearly how one PCR reactions are split in 20,000 oil droplets. These would be amplified using PCR and then be read and analyzed through color coding. And this color coding you can see clearly over here. For instance, if the FAM probe binds, and the X probe binds, it's a wild type sequence. If the FAM probe binds, it's an NHEJ template. If the HEX probe would bind, that means it's an HDR template. And now to my human colonic experiments as well as for mice experiments. As you see with human colonic experiments, you can tell clearly that there's that first for over a 5-week period how this would be analyzed. Organoids would be collected from patients, and then they would be highlighted for GFP to see whether if the treatment actually went in or not. This would be followed with the treatment re-injected in the video for this to occur. This would be followed by verification of genome editing through Western blot analysis. In mice, there would be the CRISPR-Cas9 and nickase system be injected intralectally, and this would enter the crypt cells, followed by nuclear translocation and also by different types of DNA to RNA to transcription. You can tell clearly that the normal MLH1 and MSH2 proteins would be produced, and the hope is that the mismatch pathway would be restored. So now to some limitations in future directions. So there's the off-target gene editing is present by the Cas9 nickase system. And this, however, means that it can bind into different locations other than the target sequence. This was minimized by the gRNA software simulations to ensure that there would be lesser off-target effects. There are 4 plasmids in being introduced to target cells, raising co-transduction efficiency concerns. This treatment is also only useful in patients that are being targeted in a specific region, but the treatment can be tailored to each individual patient. This improves the lives of HNPCC patients dramatically compared to colectomy, because colectomy forces patients to be dependent on parenteral or intravenous nutrition at a young age for the rest of their life. This also promotes the design of personalized medicine as well. And the reason why this is because it's only the guide RNA that needs to be changed for it to be personal to each patient. Here are my references, and thank you so much for listening.
Great job, Shravan. Thanks. And so we have a question in the Q&A. If you have other questions, feel free to throw them in them. This comes from Jin Zhao, Polygence co-founder. So she's wondering, what's next for you, Shravan? Will you be running an actual lab experiment for the experiment that you mentioned? You're doing such advanced work, super impressive.
Yeah, so most likely I plan to perform maybe in the future actual lab experiments like relating to this, and probably find ways to execute this experiment in the future.
Awesome. We also had a mentor that reviewed your work leave a question too, and their question was, how viable of a treatment do you think something like this will be, or will there be other limitations to this kind of technology, like prohibitive access because it's such a costly kind of treatment?
Yes, so in terms of viability, it is a very viable treatment. Yes, it will be possible if analyzed for limitations, things like that. But yes, it is a relatively new technology, so there will be very much limitations and things like that. Since that is there, there is a price point that is present over in this for this treatment as well, considering that, which may also be a limitation, considering it's limiting access to for other people to achieve this treatment. But the hope is that to find new ways to ensure for equity and try to make this treatment less expensive and more so to be given to multiple backgrounds of people.